So welcome to the 2026 Utah Pollinator Pursuit volunteer training. We are so excited to have you here today and just talk about how you can get involved in pollinator conservation through community science. On the agenda this afternoon, we are going to introduce the Utah Pollinator Pursuit team. We're going to talk about some of the program successes, how you can participate, some field trip options. We'll go through frequently asked questions and do a quick Q&A. I will be keeping an eye on the chat as well as the Q&A throughout the program while Mary and Amanda present, so feel free to throw any questions in there and we'll either answer them right away or we will talk about them at the end. And just a little bit about Utah Pollinator Pursuit. This is a collaborative program that is hosted between Utah State University's Rare Insect Conservation Program and Sageland Collaborative, and it is made possible through a species protection account grant through the Utah Division of Wildlife Resources. Introducing the team, I will go ahead and introduce myself. My name is Sierra Hastings. I'm the Communications Director at Sageland Collaborative. Pass it over to Mary.
I'm Mary Pendergast, one of the ecologists at Sageland Collaborative.
I'm Amanda Barth. I am the Rare Insect Conservation Coordinator for the state of Utah, but I work for USU and I work with the Division of Wildlife Resources. So I'm kind of a product of a partnership between the two.
Awesome. Thanks everybody for being here. Thanks for kicking us off, Sierra. We always like to start off with a land acknowledgement. So those of us that are based in the state of Utah, we live, work, and recreate on the ancestral and traditional lands of the Ute, Goshute, Paiute, and Shoshone, and want to offer this land acknowledgement to introduction. For those of you who don't know Sageland, we focus on conservation, community, and people. We do this through partnerships and collaboration, bringing objective science into the on-the-ground action, like habitat restoration, for example, and by working with the entire community whenever possible. So often at Sageland, we're collaborating with academic and government partners to fill basic data gaps on where species are distributed and what condition those associated habitats are in. So in some cases, the work lends itself to engaging community scientists when we can use survey protocols that can be standardized and provided in a form of a public training. So before we get into that training today, I wanted to note that this presentation is going to be recorded, so you can always go back and review it. And for anyone who has registered for the live webinar, you're already automatically going to receive emails about the project. So for folks watching this as a recording, you can go ahead and scan that QR code here to get signed up for the project newsletters. So just pause if anybody wants to grab that. And with that, I want to kick it off to our crucial partner, Utah's Rare Insect Conservation Coordinator, Amanda Barth.
That's me. Hi. So the Utah Pollinator Pursuit project was started in 2019. And this was originally launched by through a partnership with Minnie Wheeler, who is the state Utah. She's the Utah Plant Conservation Coordinator, and with Sageland Collaborative. And this initial focus was on documenting monarch butterflies and their milkweed host plants that were present across the state of Utah. And then the following year, we expanded our efforts to, I joined and we expanded our efforts to bumblebees. So that was 2020. And then we added habitat assessments in the next year. And then we expanded and changed to collecting information on all insect pollinators, as we saw this incredible potential for the power of community science in the state. So some highlights of our huge successes in this project: we have now over 1,800 species detected of different pollinators in the state, and that's including bees, butterflies, flies, moths, wasps, beetles, and even thrips. And we have engaged hundreds and hundreds of community scientists, many volunteers who have joined us through trainings like this. And we have benefited from more than 2,500 hours of time that have been contributed by volunteers on by year per year. We have more than 84,000 observations in our combined databases between iNaturalist and our previous data collection platform in Survey 123. So 84,000 pollinator observations in 6 years, because we're launching into our 7th year, is pretty good. That's a pretty great way to stretch the dollar. We have a lot of amazing partners, including our the Division of Wildlife Resources. I am just stuttering. Fish and Wildlife, BLM, the Forest Service, NRCS, National Park Service, and so many local groups around the state. So again, we have over 1,800 pollinating insect species detected. And of course, that doesn't mean that that's all we have. We have this is informing our checklist of pollinating insect species that occur in the state. And the community science data contributed is helping us understand species ranges and phenology, the timing of their activities, the relationships that they have with plants, their own metamorphic stages, and a lot of other really fascinating things that it's just a huge, really invaluable wealth of data. So if you happen to see any kind of insect on a plant, on a flower, or plant, period, or any insect, but especially those visiting flowers, you can consider that a pollinator. So if you can get photos of it, submit your best photos to iNaturalist, and that counts as an observation. And then you're helping advance our knowledge of that species in the state. So part of the applications in the last 6 years of the data that we've collected through this project the data have enabled us to, with the Rare Insect Conservation Program, to assess the conservation needs for dozens of species and identify several at-risk insect pollinator species that we have deemed that meet the criteria for species of greatest conservation need. That's the acronym is SGCN in the state agency world, and that list of SGCNs is what we shaped the 2025 iteration of Utah's Wildlife Action Plan, which is a whole big conservation plan with guidelines for conservation of all these at-risk species and their habitats to prevent their potential listing on the endangered species list, and also to protect them from any kind of risk of extinction. So 11 species of pollinators already is fantastic, and that it's really quite a data demanding process to assess those things. So I'm just trying to point out how incredibly valuable this data set has been for our program. It can also be really challenging to prioritize conservation efforts without knowing where those efforts need to be focused. And one of our key species, well, several of our key species, especially in these 11 species, is monarch butterflies. And it has been a top priority since the inception of this project. Just in this last year, we had volunteers submit 261 observations of monarchs that were confirmed. We had observations submitted from all 29 counties across the state. And that's really saying something, because the majority of our population lives in Salt Lake, Summit, Utah County, Washington County, Cache County, like along the Wasatch Front. So, but that's the majority. That's about 3 million people, but there's still coverage across the entire state. So that's really significant, and that's an incredible success story for our volunteers. In past years, we've only had, well, not only, in past years, we've had several hundred bumblebee observations submitted. In 2025, UPP volunteers submitted more than 3,000 observations of bumblebees. So you guys are really looking for them and you know how to photograph them, and that is really something. And again, we have, just in this past year, over 2,500 hours of volunteer time, and that's assuming that we have 1 hour per volunteer observation. So in just in this monarch specific role, this little story map kind of shows you it's a compilation of, I lost my notes here, compilation of data showing monarch observations at different life stages and sort of where those densities occur. And so again, you can see some of these areas of a lot of large urban centers tend to be where we see a lot of monarchs as well. But out in the Uinta Basin, there's also a ton of observations that pop up at certain points in the year. And this, hopefully you can see the little icons. Some of them are eggs, some are caterpillars, some are pupae. This kind of information is really, really valuable for us to understand where monarchs are at different points in the year in Utah during their breeding season, and how they are using that habitat, and just like, are they breeding? If you're seeing eggs, then they definitely are. So this is really valuable. And so one other really cool thing that I want to share is these are two examples of habitat distribution models that my team, Adam Savinsky and Iris McCulloch, worked on this past year using Utah Pollinator Pursuit data submitted by our volunteers to really come up with really, really interesting and beautiful models of where potential suitable habitat is across the state. So this is really helpful for us to both survey those landscapes and find populations that we might not know about. And also it helps inform us in the restoration efforts that Sageland is going to talk about next. But this is the kind of thing that really is valuable. And we did not have this kind of information. We did not have these kinds of models prior to last year and Adam and Iris's work. And we didn't have the data to inform those models without Utah Pollinator Pursuit volunteers.
Thank you so much, Amanda. It just means a lot to hear from you as the state, rare insect coordinator, how valuable this project is. So thanks for taking the time to share some of the highlights.
And we've only found a few ways to use the data. It is such a huge wealth it's a treasure trove of really incredible information. And it means more than we have even begun to grasp.
Thank you. So yeah, having gathered lots of data over the past several years, we now have that information on a few of those especially rapidly declining species to get to start targeting habitat restoration efforts on the landscape where we think we can have the most impactful conservation outcomes, especially for those pollinator insect and plant communities. But as we know, all those things are connected. So if you've got the plants supporting the insects, you've got the insects supporting the bird communities. If you've got those freshwater plants and freshwater insects, you're supporting amphibians and fish and other aquatic species. So it's obviously all very connected. And that's not even to mention how valuable pollinators are for every third bite of food that we have, right? So many reasons why these data are going to be useful to targeting, restoring functional habitats. So this year, we're continuing to ask community scientists to collect pollinator observations, as conservation is an iterative process whereby we gather information, plan the conservation action, work to restore the habitat on the ground, and then continue to gather those data to measure the plant and insect responses to the restoration efforts so we can adjust accordingly. So please continue to help us put pollinators on the map by participating in the iNaturalist training today and this field season. And also, please keep an eye out on your emails from Sageland Collaborative for ways to participate in pollinator habitat restoration projects on the ground. So we can put these data that you're helping gather into conservation action. So we'll talk more about specific opportunities to get in the field at the end. So with that, we'll move into how to participate in this project and actually add pollinator data into iNaturalist. So first, a quick note on safety. We ask that you agree to work safely because your safety is our number one concern. We want you to stop surveying anytime there's a concern for safety. Never survey alone. Partners are encouraged. If you see something, say it, and watch out for each other. In addition to your physical safety, we care deeply about your well-being. We ask everyone to treat each other's unique perspectives and identities with respect. If you have any questions about our culture of inclusion at Sageland, please reach out personally or anonymously through our webpage. So some of the most common hazards of this project we're talking about today include just slips, trips, and falls. So be really mindful where you're stepping and take your time as you walk around. Be prepared with suitable footwear. For all of you who like to get up close and personal, like most of us, when taking an image of a pollinator, be careful not to poke yourself in the eye as you move closer to the vegetation. This can be a really common injury as you're bending over to focus on photographing an insect, and then you're not noticing that you're getting closer and closer to that vertical vegetation. So I myself have put my sunglasses up to get that great photo and just nailed my eyeball with, a piece of vertical vegetation in a time or two. So that is probably the most common injury. So keep those sunglasses on or just pay attention while you're focusing on taking those pictures. So to participate in this effort, all you need to do is download iNaturalist on your device and search for the Utah Pollinator Pursuit project within the iNaturalist app and select the Join Project button. If you can, please also take that additional step once you've joined the project. There is a setting for your membership setting button, and you go in there and select Trust Project Administrator option. So that will allow us to see the exact location of where you took photos and insects. And in particular, this will allow Amanda and other state biologists to get the most detailed information from your photos while still keeping those exact locations masked from the public. So once you have the iNaturalist application, it's as simple as selecting that plus button to add an observation into the project. And you can, from there, take a photo through the app itself by clicking the Take a Photo button, or you can select an image that you've already taken that is saved on your device. So when you are observing pollinators, normally an insect on a flowering plant or an insect flying around flowering plants, it can be tricky to get a clear image that allow you or others using the application to positively identify the species. The good news is you don't need to know the species you're taking a photo of to get a really quality image or images that other iNaturalist users and insect experts can eventually identify. So here are some tips for photographing. We really want to see the flowering plant, if possible, in the photo. A crisp side view can really help us make out the different body segments, as well as the head and antenna, which are often distinguishing features. And an image where the eyes are in focus is also key for some identifications, as well as a dorsal view of the abdomen or the wing patterns. Also, if you see pollinators on the move, don't hesitate to just start taking video of your subject as it's flying around the flowering plant or the nectar source, rather than trying to wait for the perfect moment for a still photo, which may never happen. You can later select multiple frames from the video that you've taken and choose the clearest images for your iNaturalist observation. So now, if I'm seeing lots of activity, bumblebees or monarch or whatever flying around, rather than, following it to wait for it to land on something, I just start taking video, and then you can go back in and select where you have a frame of a clear view of those anatomical features we just talked about. So now you can start logging pollinator observations anytime on your own, whenever you like. You can also view and enjoy the images and observations of others in that project or throughout iNaturalist. We also want to make sure that you are aware that you can check out monthly highlights through the project journal, so you can review those within iNaturalist. You can learn things through the journal about what species of conservation need have been observed and who the top observers were that month. Maybe you will be one of them in the future. Also, stay tuned for in-person events where you can pick up this year's Pollinator Pursuit sticker, which is so beautiful, created by Sageland's very own outreach coordinator, Francis Go. We're excited to share some upcoming field trip opportunities with you. If you want to go beyond logging observations on your own, you can join us on some guided surveys where we will be looking at areas where Sageland and our partners have already conducted some habitat restoration activities, like installing human-made beaver dams and native pollinator plants. So during these field trips, you can get some hands-on experience using iNaturalist, and you can help monitor pollinator insect presence around these restoration sites. So if you haven't already, be sure to sign up for Pollinator Pursuit's newsletter so you can get all the details on how to sign up for these future events. I'm just going to pause. I think we've shown that a couple times, but just in case people need to grab it, that QR code. Additionally, Sageland's outreach team will be hosting 3 different bug club evenings in more urban locations around the greater Salt Lake area this summer, where you can learn about identification and photographing pollinators and work in an outdoor group setting. If you sign up for the project newsletters, you'll also receive information on native plant and seed swaps and sales and other ways to get free plant materials. We want to share one of our partner programs as well. The Xerces Society for Invertebrate Conservation hosts the Bumblebee Atlas training, where you can then adopt a grid cell in your state or a neighboring state and conduct dedicated bumblebee specific surveys. This project also provides essential data for creating evidence-based conservation plans to protect these declining pollinators. A specific Bumblebee Atlas opportunity is coming up in May in Kanab and Escalante, Utah. So I'll just pause again so folks can grab that QR code for the Bumblebee Atlas field trip. And if you want to learn more about Bumblebee Atlas, you can get there through that same code as well. And with that, I'm going to kick it off to Amanda. We have a recent addition for another project here that we'd love to hear more about.
Thanks. Yeah, so another really very cool and like cutting edge way to support monarch conservation is called Project Monarch. And so there's an app associated with that because we have no shortage of apps in this world. But this app allows the public to track migrating monarchs who've been tagged with little teeny tiny solar powered transmitters. And in that photo on the right, you can see that little monarch has a tiny little thing glued to his thorax with a little antenna extending backwards over his abdomen. So those are called blue morpho tags, and they were developed by the cellular tracking technology CCT, and they have deployed these tags all over the country, and they are giving people the ability to use their phones to detect these tags that have Bluetooth frequency, and follow individual butterflies on their paths. So if you happen to be within a certain distance of any monarch that has that tag on it, your phone acts as like a receiving tower, and you can submit your data to the project. So this is huge. Of course, we have more QR codes for that as well, but so the folks in Utah, if participating in this, would be complementing the really invaluable tagging efforts that are being done by a bunch of fantastic biologists out in the Uinta Basin who have been tagging monarchs for years through some other projects. But this one is giving us much more real-time pathway data And their tagged adults in the past have been detected in Arizona, in New Mexico, and in California. So this shows just how incredibly important Utah breeding grounds are for the Western migratory population. And so this is one of the most exciting new technology projects that is helping to answer some questions about where the migratory paths are in Utah, and also where we can again target our restoration efforts. So it's a big one.
That's awesome. Thanks, Amanda, for sharing the background on that. So in review, we have some frequently asked questions from our volunteers before we open it up to you all for the live questions. So who can participate in the project? Anyone is welcome. Please share this with friends, family, coworkers who might be interested, and get signed up to receive emails about more specific field trip opportunities. What species are we looking for? Please take photos of any insect species you see on flowering plants or around different plants. And make sure that your photo georeference location setting is on so that you can add that photo later to iNaturalist, and the location will be attached to it, so that you could also add that manually if you needed to do it after the fact and not when you're real time, maybe in low service. You can always be on the lookout for insects when you're out and about at work or recreating outside. So again, if you see an insect on a flower, log it. If you see it on a plant, log it. If you see it flying around a pollen or nectar resource, log it. So where do I enter these data? So for any pollinator insect you see or just happen to see when you're out and about, you don't have to be on a field trip or in a special designated area. You can be in your neighborhood, your own backyard, at the park with your family. If you see it, you can log it in the iNaturalist application, and we hope that you take that additional step of joining the Utah Pollinator Pursuit project. Remember that these data that you help us gather about pollinator insects allows us to prioritize where habitat protections or restoration activities should be taking place as we engage in conservation planning for these often overlooked species. And with that, I think we'll check in with Sierra on questions.
Awesome. Thank you so much, Mary and Amanda. We've got a couple of questions to start off. Klaus was wondering if we integrate data from the Bumblebee Atlas.
Thanks, Klaus. That's a great question. We do. We try not to overlap the two data sets. So, but Xerxes communicates with me directly, and so I get their cleaned up data set so that my program would enter it directly into our state heritage database. So it's not duplicated and being, yes, we just don't want to have duplicates. It saves us a lot of time and headaches. But yes, we do get the data, and they get our data as well.
Thank you, Amanda. And just to clarify, as somebody who has also done Bumblebee Atlas, you should not log the same bumblebees that you logged with Bumblebee Atlas in iNaturalist. Correct?
Yes, just to clarify, the surveys that you're doing, your dedicated bumblebee surveys, those are Bumblebee Watch data. That's what you submit to Xerxes for the Mountain States Bumblebee Atlas project. If you are out and about and you're not doing a dedicated survey and you're not at your assigned grid cell that you've adopted, iNaturalist is your way to go. And that goes for everything that you see. So we call those incidental or opportunistic sightings. But if you are out doing a specific data collection and you're timing your survey and you're catching your bees and doing that regimented protocol, that's Bumblebee Watch territory. So we don't want those data yet. give them a Bumblebee Watch.
Perfect. Thank you.
That's a great question.
It is a great question. And we do love our Bumblebee Watch, Bumblebee Atlas partners. Vanessa is asking, regarding monarch butterflies, they've heard conflicting information regarding butterfly memorial release events and the disruption on migration and with other wild butterfly habitats. Do these farmed monarch butterflies hurt conservation efforts?
So uh, we in the Western monarch conservation world that I am a part of, we do not consider captive reared monarchs a viable conservation strategy for the Western migratory population because there are some concerns about the facilities and the materials that those monarchs may be reared in that might not have been sterilized, and there could be the potential for transfer and accumulation of OE. And when you just have a lot of something in a certain area, it tends to be a sink of disease and parasites. And so unfortunately you're also releasing individuals into the wild that have been more or less acclimated to artificial environments, artificial conditions. So they might not really be able to cut it in the temperatures that are, much more extreme than a more controlled environment, say in someone's basement or bathroom or like living room or something, or just in a greenhouse or wherever someone's raising organisms. Not to say that there aren't successful captive rearing programs for butterflies, but monarch butterflies are very beautiful and they're iconic, and people love to see them fly away, and so and there's a lot of strong feelings about being able to share that experience with people, and there is not any regulation, and we don't have the ability to regulate monarch rearing and handling in the state of Utah. But it is also illegal to ship them across state lines. And so other departments of agriculture are, and the federal APHIS, the regulatory arm of USDA, they're very concerned about insects being moved across state lines, even if they're native. So yeah, it's also possible that when you have these monarchs to release, that they were not necessarily sourced from the local gene pool. They could be from the eastern population, and they have different traits, slightly different traits. They're not a different species, but it's just having you're setting them up for failure if they are released into an environment that they really aren't adapted to. If that is, hopefully it makes sense.
Yeah, thank you. It's such a complex issue.
The thing we can really focus on is documenting their presence and putting a lot of energy into creating habitat that's healthy for them, because the reason for monarchs decline is a loss of available and suitable habitat. It's not because they are somehow not able to survive from egg to adult. It is that they are at their carrying capacity threshold, and the ecosystem cannot support a larger population because habitat loss and temperature and extreme temperatures and drought and pesticide presence. So those are the things that are causing the decline. Those are the things that we should be targeting to increase the population.
Amazing. Thank you. And then we have another question from Klaus about honeybees.
We don't want them. Nope. Thank you. Thanks, Klaus. Honeybees are like, they're agriculture, and people are submitting plenty of honeybee observations to INET, and I have them specifically cut out of the parameters for the collection project. So we don't need honeybee information. And if I need the information, I can find it, but I'm not looking for honeybee presence because they're not native species. They are managed, and if they are found out in public lands, they were either put there by they're coming from a managed hive, or they are potentially a feral colony, and that's more of an agriculture, a UDAF kind of territory, and not for me.
That being said, if you're new to the project and new to pollinators and you're like, I'm not going to know a honeybee from other native bees,
sure,
go ahead and take the photo. Like you will not be penalized. We'll still take those data. And as Amanda said, she's able to filter those out. So that's really the beauty of using iNaturalist is like send us all your photos, and we'll be able to sort out, the species that we're really focused on. So don't let your inability to identify a particular species stop you from submitting a photo. Yeah.
Also, I have taken plenty of really beautiful photos of honeybees on my garden, so I think they're very sweet little bees, but that's not what I'm collecting information on. So, but go ahead and get the practice in, admire their gentle behavior,
Thank you both. We have a great comment from Natalie in the chat that says that they're like tracking chickens instead of bird watching. Do observations.
Chickens are cute.
They are cute.
We like chickens.
Yeah. Another question from Leanna. If we use Seek, will that matter or impact the project, or does it not matter?
So my understanding with Seek is that it is like an initiative that iNaturalist launched, but is no longer funding updates for. And I don't think that, and forgive me, I have not even explored Seek as an option. It's great for identifying things, but I don't think that it is a portal for data to come into our project. But I should probably look, but I don't think that it has been updated, and it's been several years since they've updated, the project. It's that initiative.
Yeah.
It's still useful. So you can't, but it's useful for like users. It's not necessarily useful for the project manager like me.
Yeah. I will say iNaturalist underwent a upgrade last year, and that's the QR code that we link to now, is the newest version of the app, and it is much more user-friendly. You are able to access the project through the app now, as well as look at users and do all the fun things that you can do on the web browser. So definitely, if you were wary of iNaturalist before, give it a try now. Natalie asks about observations made on iNaturalist. They may have a data quality score. It will show a green check for casual grade, a green check for needs ID, and then a gray shadow for for research grade. Is it okay that their observations aren't research grade, or are there additional steps or information needed?
So that's a really wonderful question. The research grade status for data is a goal, but it's also very fluid, because it basically just means that at least two users on iNat agree with a species identification. And that can be finicky because sometimes there's, inactive taxa, and then they change names and whatever. And there's also some reevaluation of taxonomy happening. And so people, someone who is doing a deep dive and they are an expert in a certain, maybe they're looking at leafcutter bees in particular, or someone is right now doing a very thorough reexamination of digger bees, and she's going through digger bee observations across the West and either giving specific identifications to things that were just at a genus level, or saying, this was identified to a species level, but actually we can't identify it. So, they suggest a genus level instead, because there's really not enough certainty. It's hard to identify species from photos. It's not foolproof. It's like I said, it's fluid. So my own personal observations, I probably have about 50% of my observations at search grade, which is, it's nice. It's kind of like a dopamine kick, but it's not necessary. It is really valuable. And again, you might know exactly what a species is, and I might know exactly what a species is because I've had a conversation with an expert about it at, some meeting, or I've read the paper, and there are not other people on iNaturalist who know that information or have ever seen it, or ever seen my observation. So it will just kind of linger, or, it may stay as, needs ID for a long time. And maybe 5, 6 years down the road, someone will come back and they're doing their deep dive and they're like, I know exactly what that is. And I've been identifying these things all week. And so it's fine. Basically, long TLDR, it's fine if they're just, whatever you submit is valuable, and it's adding to a massive data set that is going, the possibilities are really endless. So it's fine.
It's fine. This is what I tell myself too when my favorite observation does not get to research grade. It's fine.
Yeah. And I will warn you though, that there are a couple people in the INAT community who are really brilliant experts, but they're not very, very nice. Don't take that personally. They like to be right. And it is not about, it is not about you being not informed on exactly what a species is, or going with the machine bit, the machine vision, the computer vision identification. Like if you made a mistake and said it was a fly and it was actually a bee, whatever, and they have something snotty to say, don't take that personally. And I apologize in advance. I can't help that. I do scold some of those people, but it is the way in these social communities, like these online communities.
It is. All we can do is be kind.
Be kind and be humble.
Yes. Another great question from Klaus. If you are out and about and you see many unique individuals of the same species in the same location, same day, should you try and capture all of those as a unique observation?
That's a good question too. I think that if you are planning on submitting, and if you want to photograph several individuals and submit them, if between observations, try to like take a photo of something else so that you kind of have like a bookmark or a place marker between individual, like groups of photos of an individual, so that you can just submit photos of a single individual at a time, if that makes sense. So you might have 8 photos of one bee and maybe 2 of a separate bee, and a couple more of another bee, but they're all the same species allegedly, or potentially, ostensibly is probably what I want to say. But there could be different features about those different individuals. They might actually be separate species. There could be one that has a mite or a parasite or something else, interesting, some other feature. And Klaus, I know you take really good detailed shots. So being able to zoom in on those images and like notice that there happens to be, some sort of abdominal parasite peeking out through a tergal segment is really cool. And that's, it's best to have that be an individual observation so you can refer to it. So yeah, that makes it really valuable data that way. But don't try to try to get every single individual that you see out in a certain spot, especially if there's a lot of them, because the using community science data to try and estimate populations is kind of, it's one of the limitations of this kind of data. So I wouldn't say that that's like, don't challenge yourself to do that, because that is not what that's not the question we're trying to answer using this information.
Yeah.
But keep taking amazing photos, Klaus.
I do believe Klaus is one of our top observers. He is our number 2 top observer
year after year. Good job, Klaus.
So Klaus, keep it up. Thank you. If there are any other questions, now is the time to pop them in the chat. The OCD does not go that far.
Good.
That is good. One comment I will make is that you may have noticed a lot of insect photos throughout this presentation, and many of those are pulled from iNaturalist. We are so grateful to everyone who does allow copyright use of their photos for nonprofits, NGOs, other organizations, and we are so grateful. If you are able to check and just make sure that you are sharing your photos, that is just a great setting in iNaturalist. It's the CCOBYN feel free to message us if you're not sure what that looks like. I think it is the default setting, but it allows us to use your photos for education and for outreach.
Never sell them.
Never. We
No.
never sell them. We
Or
just
plagiarize,
feature
which
them.
never.
No. And we always share with credit because y'all are amazing artists.
And AI sucks. No, 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 no.
All right. We would never give them to AI.
Never.
Never. All right. If there's oh, Natalie asks, who runs the Pollinator Pursuit Instagram page? I do believe that person is here with us today.
Yeah, you're
It's
welcome.
Amanda.
Sometimes I neglect it because it is social media and it is a lot of it is a time suck. But there's a lot of other cool accounts that I can constantly, regurgitate,
Yeah.
recycle for people. You're absolutely welcome. Thank you for following.
All right. If that is the last of our questions, we'll go ahead and wrap up. Mary, do you have any final thoughts?
I would say just my final thought is if you really are interested, well, if you are interested in coming out with us, we're excited about this new opportunity to do some monitoring at those restored areas. And this is kind of a pilot effort. We're going to see how it goes this year. And then we hope to try and do this more with some of Sageland's other restored habitat projects. So, as they are put in and then as they are established, we will probably see a change in the cast of characters in the pollinator community in those locations. So it's you get to help us come out. You get to come BioBlitz these sites with us and frolic through a beautiful landscape and for science. So it's fun and it's healthy and just all around good time.
Yeah, you nailed it. That was the other thing I was going to add, and just yeah, the folks who have participated in the past know how rewarding this kind of stuff is, and it's also just a really fun time. And we have food, and it's just a great group of folks. There's self-selecting. It just ends up being a great time. The group that turns up for these things. So just really appreciate everybody who's done it in the past. And if you're new, just know that it's a good time.